science seminar, the first of the fall. Today we have Sarah Alamdari, who is a senior applied scientist at Microsoft Research for the last 3 years or so. Prior to this, she finished her PhD in chemical engineering from the University of Washington, working with Jen Fender. And we're very excited that she's here today to tell us about generative AI and protein design. So thank you, Sarah, and the floor is yours.
Awesome. Thanks for that intro, and feel free to interrupt with questions as I go. But yeah, let's kick it off. So a lot of our work currently is motivated by this idea that protein design can unlock new biological and therapeutic capabilities. Thinking about things like impacting medicine, tech, pharmaceuticals, and beyond. So, for example, we can create new molecular tools, we can engineer enzymes to perform new chemistries, and we can even design new therapeutics. Now, all of this chemical diversity that we observe in nature arises from 20 distinct amino acids. In 1965, Margaret O. Dayhoff published this atlas of protein sequence and structure, where she collated all the 65 proteins that have been known at the time and introduced this single letter amino acid code that we still use today. And this describes all the unique chemistry in these amino acids. I'm sure everyone here is familiar with this, but this is the structure of an amino acid, where that R group side chain can have a unique chemistry, as shown here. Now, this sequence structure function paradigm is this idea that protein function is encoded by a biological language. So given a sequence of amino acids, this is what encodes a protein structure, and together this sequence and structure enable a protein's function. This begs this question: can we learn this paradigm in order to design new functional proteins by understanding this relationship between sequence and structure? Now, there's been a number of recent generative models that have been focused on achieving new function through the design of structure-based approaches. Now, this is an approach that's been incredibly successful. Methods like RF diffusion have really taken off in this field and catapulted it forward. But interestingly, these models often require this intermediary step of designing a new sequence that fits these structural constraints. Now, we thought, instead, can we just treat sequential design space and design protein function directly in this modality? So in addition, we're proposing sequence as this universal design space. We're also motivated to eliminate this need for indirect sequence design, as I just highlighted. Design for proteins that might fall outside of this structure function paradigm. In a really dramatic case, this is characterized by these intrinsically disordered functions that don't have a characterized or well-known secondary structure. And lastly, although these protein structure databases are incredibly rich and high quality and contain hundreds of thousands of proteins, our last motivation was to take advantage of the massive amount of scale that occurs in these protein sequence data sets. They're orders of magnitude larger than anything you can obtain in structure space. These evolutionary scales of data also span across multiple organisms and many novel functions. So to that end, I'm going to cover our team's work on 2 of our latest projects. The first is EvoDiff. It's an evolutionary-inspired discrete diffusion model. And the second is Dayhoff, a follow-up on this work, a tribute to the work of Margaret Dayhoff, and it's a new and updated atlas of data and models for protein generation. We'll start with EvoDiff. So Evo here is indicative of evolutionary scale of data, and diff here is referring to the diffusion modeling approach that we used. And before I go on to describe the results, I want to take a second to acknowledge the entire team who made these next slides possible, many of which who are at Microsoft: myself, Rianne, Neil, Nicolo, Ava, and Kevin, as well as Nitya, who worked on this project while an intern at MSR a few years ago, is now a PhD student at Stanford, as well as Alan and Bob, can't see my mouse, who are highlighted here. These are our collaborative experts on intrinsically disordered proteins at the University of Toronto. Now, briefly, EvoDiff is trained by modeling millions of sequences from UNIREF 50 over a discrete diffusion process. What that means is that going from left to right, we iteratively corrupt an input sequence. Nope, going from right to left, we iteratively corrupt an input sequence over each position in the sequence fully corrupted at time t. And at each step of this model, the model is tasked with predicting those masked residues at each time step. Like I said, we trained this over 50 million sequences in UniRef, and the backbone architecture is a 640 million convolutional neural network. Now, if you're familiar with generative discrete modeling approaches, EvoDiff generalizes both masked and autoregressive language models, meaning that we're modeling every sequence order at every step. Compared to a left to right autoregressive model, it's only modeling one decoding order at a time. So it's always going from left to right. And I'll showcase this later, but the reason we chose OADM or wanted to pursue this modeling approach is that it allows for conditional generation, meaning you can fix or mask out part of the sequence and then in-paint that with the context of the rest of the sequence. This also generalizes concepts from masked language models, which are just modeling a fixed step of this process, so a fixed number of masked residues at train time. And in practice, we see that this order-agnostic diffusion method generalizes better when being tasked to generate new proteins from scratch. Okay, now that we've had a trained model, at inference, we can now from left to right start with a all-masked sequence and then iteratively decode randomly at every position what amino acid should be in there. We're only doing this in sequence space, but on the right, I have a schematic of what this structure is doing over time in this process. But this is just a predicted structure. Okay, so how do we evaluate the outputs of this model? Now, to evaluate the quality of what we're calling these unconditional designs, we compute a number of in silico metrics. Namely, I'm going to focus on this pipeline here, where we start by generating a sequence, input it into a structure prediction method. In this case, we use OmegaFold. We then inverse fold that generated structure and get a new predicted sequence and compute the self-consistency perplexity between the original sequence and the one that's predicted using this third model. We can plot that metric here on the left, where going down is giving you better self-consistency metrics, and going right is giving you higher or a better structure prediction confidence metric. You can see that the range of these values on natural proteins or our test sequences are quite diverse, but generally we categorize things as being in that lower right quadrant as sequences we're fairly confident in. So we can do this for a bunch of EvoDiff generations, and while we don't do as well as natural proteins, we can generate enough things in that quadrant to pull out and look at. So here are a few examples that I pulled from the paper of sequences that have relatively high scores and low self-consistency. They have a range of various coverages and identity to natural sequences, so they're not truly de novo in the sense of that. But we're able to get 20 examples of this, 25 actually, and express them in E. coli using no filtering criteria besides what I've described here. We send these off to a wet lab, and we find that these 4 examples in particular that I pulled expressed and have some elements of secondary structure as given by the circular dichroism spectra here. They don't do great in terms of comparing against their predicted secondary structure, but we're able to make these in a lab. Now, here's a second metric that we're using to evaluate the feasibility of these sequences. We take a set of test sequences, we pass them in a model PROT5 and get an embedding space, and we can map that using TSNE in 2 dimensions. We then do the same exercise with a set of generated sequences. We get another set of embeddings, map these in space, and we look at the overall distribution of our generated sequence along the natural sequences. We can then calculate this with a metric we coin the Fauché protein distance, where a lower value is better here. Now, we don't do a great job of capturing all natural proteins. There's clearly some regions that are missing here, but we can compare this to some other baseline models. If we compare to a left to a right autoregressive language model, we see that that model does quite a bit better in this task and a few other ones. But remember that EvoDiff enables that idea of conditional generation. We also compare it to embeddings from sequences generated from ESM2, and we see that EvoDiff achieves higher quality generations. And lastly, we compare it to structure-based diffusion, and we see that the coverage over natural sequence space is much larger than compared to a model that has only been trained on structured data, indicating to us that EvoDiff is learning something from this evolutionary scale data. Now, EvoDiff's OADM diffusion framework introduces this natural method for conditional sequence generation that I've been alluding to, where we can fix some set of the subsequence and then in-paint the remainder. Now, because the model is trained to generate proteins with this arbitrary decoding order, this is easily accomplished by just masking and decoding the desired portions of your conditionally designed sequence. Before I get to that, I want to introduce the second model that we trained in the paper. It's called EvoDiff MSA. It's better at leveraging this evolutionary information. So an MSA, or a multiple sequence alignment, is capturing these evolutionary relationships by aligning sequences to a query sequence. In our case, we use a database that had constructed some homology search to construct these MSAs. We subsample them down to 64 sequences, and we have a max context length here of 512 amino acids. Now, we train this model using the exact same task that we used in sequence space, but now over an entire MSA. So that first row is always the query, and we iteratively mask at each portion in the alignment. Like I said, this is trained over 100,000 MSAs. They're taken from this OpenFold database, covering, again, many organisms and functions. This time we use a different architecture. This is one that's built to handle, or had been designed to handle multiple sequence alignments. It's called the MSA transformer. And this came out of Meta back in the day. Now, what's cool about this is we've really trained this amortized local generative model where we can design the guide of proteins with supplying it on sets of related proteins here. At inference, what this looks like is masking out a query of a protein that you have a desired function for or a desired MSA, and then using that model to generate a new query sequence, given that context of those homologous proteins. Now, we validate our model in a few different ways. I forget which ones I include in the slide, so I'll put them all here. The first is repeating that first exercise we did for sequence only, where we run them through this fidelity pipeline. And overall, we see that the MSA generated designs are a much stronger baseline than just using a sequence only example. We then in-paint some sequences using IDRs as an example. We do some work on functional scaffolding. And we're going to find out which ones I included. Okay, so it looks like we're going to talk about the latter two today. So let's start with sequence in-painting. So as I alluded to earlier, not every protein folds into these static 3D structures. And in many cases, for many people in this room who work on MD, we know that in general, proteins are not static structures. And in these cases, structure-based design is just not a viable example because this function is not mediated by a structure. Now, this case was particularly interesting to us because we know that structure-based methods tend to overproduce these helical or structured components. So this is a graph of the distribution of strand and helix regions that are predicted to come out of generations from EvoDiff-seq versus the comparable structural model RF diffusion. Alternatively, we see that in this region where that structure is missing in RF diffusion, we have some note of that in our EvoDiff generations. Now, to try and demonstrate this, we choose a target, COX-15. This is a protein that has a mitochondrial targeting function, which relies on the presence of this disordered region at the N terminal here, from residues 1 to 45. So what we do is we employ two different strategies using both of these models. First, with EvoDiff-seq, we mask out that RDR region and then have the model generate for us some candidate IDRs. In EvoDiff MSA, we do the same thing, but now we provide it with a set of homologous sequences and ask it again to generate new IDRs for us. We play with a few different disorder predictors here. One is called, it predicts if that target has some mitochondrial function, and then one called, I believe it's called Dr. Bert, which predicts if that region is in fact disordered. Now, to evaluate this, Robert, our collaborator, which I mentioned earlier, conducts this microscopy assay in yeast to assess the cellular localization of the COX-15 proteins with the EvoDiff designed targeting signals, and compares that to COX-15 with its native IDR. He tags these generated constructs with SFGFP, which gives us a green signal, and then recombines them into a yeast strain, which expresses this endogenous COX-15 locus tagged with. And if we get an overlap between these two signals, this indicates co-localization, which is great. So now we have a visual metric if these designs are co-localizing. One assay. Yes. Yeah. So we integrate both of these into, wait, not we. Robert integrates both of these into a yeast cell. Okay, so first he images the green channel, which is going to show localization of the designed COX-15 variants. Then he images this red channel, which shows us localization of that native COX-15 tagged with mCherry. And the overlap of these two regions should give us like kind of a nice orange, which indicates subcellular localization. Now we can correlate these images with each other to quantify co-localization. And consistent with, actually not, I didn't think any of these were going to work, but in fact, every IDR that we designed exhibited a subcellular localization pattern that was consistent. Yes, so in red here. One is N1. This is one that the Moses lab had designed hoping that it would have mitochondrial activity. It passed kind of some metrics for what they thought might be a new targeting signal. They took it from another protein. That one failed here, so it's quite a hard baseline to beat. And then we also try baseline where we just like delete that altogether. This is more of a control, and that one does not localize as well. So I will say the good benchmark here for being impressed by results is that they were impressed by these results because they've tried this many times before. And so it was quite exciting to get that. Great. Okay, so that's a nice example of IDR. There's a few in silico experiments we run in the paper, and I suggest you look over them. But now we'll touch on this idea of functional scaffolding. So many of these biological interested in are mediated by this motif that is stabilized by a scaffold. And we can define these in 3D structural space. So some examples of this are active sites and enzymes, where your amino acid residues in your protein are directly coordinated with something, protein-protein interactions, which can be defined by those residues in which they interact with, or epitope presentation. Now, we had a question, kind of an ambitious question if you ask me, is that can we scaffold these using sequence information? So to do that, we take the sequence of the original protein. We pull out a functional motif that, for the case of everything I'll show here, has been pre-identified by a benchmark put together by some students in the Baker lab. We randomly place this motif within a scaffold of fixed length, and then we in-paint all the masked regions, hoping to generate a new protein that can scaffold this functional motif, keeping that functional activity in that domain. And we do this without needing any structure for functional scaffolding. We did, because we didn't want to induce like too much variation. But I actually think you could get much better results if you start to vary the distance here. Yeah. And you'll probably also pick up on is when that region between disjointed motifs is low, we often recapitulate the residues between those regions. So here's that example I've just shown of one example of a few that we decided to test in the lab. We do evaluate the things that come out of these models. Not everything's perfect. So there's two criteria here. One is the PLDDT, which is that folding confidence metric. I think we set a threshold of about 70 for as many examples as we could. We probably went a little lower. And then the second is motif RMSD. So we track the RMSD of the original motif and then the one in the folded, the predicted folded structure of the newly designed protein and hope that it retains that same shape. So this is an example of one that we tested in the lab, actually, and we see that this spectroscopic signal shifts in the presence of calcium 2+, which indicates that it is binding. We obviously don't have like specific information on where it's binding, but impressively, maybe not impressively, 1 out of the 12 designs that we sent to the lab were able to successfully bind this ion. And I think it's important to note that while we only tested, I think, 2 or 3 of our scaffolding examples, in silico metrics show that these types of approaches have complementary strengths to RF diffusion. So if we compare our best method, EvoDiff MSA, to something like RF diffusion, we see that at least from their in silico predicted success numbers, they have like quite orthogonal problems that they do well on, which is really interesting. Now, this is a weird example, but I thought it was fun to show. We include this problem where we scaffold the transactivation domain of p53, which binds MDM2. p53 is a tumor suppressor in cancer, and you want to competitively inhibit this p53 via some synthetic scaffold. This is a desirable way to inhibit this interaction. Now, unlike structure-based methods, we don't include any information about the MDM2 binder at the interface. And so we tried to just build a synthetic scaffold based off the full p53 structure, which actually is predicted by AlphaFold 2 to be like have these largely disordered regions. Whether that's its true structure or just how it behaves under these modeling conditions of this folding model is, kind of up there for debate. But we nominate a few designs. Now, in this case, we don't have that PLDDT metric to rely on because everything that comes out of this model has like poor folding predictions. We select things that kind of retain our motif and are a little bit different in identity. Now, using CRO, we were able to test 24 of our designs, and we actually find that 8 demonstrated some measurable binding affinity at varying strengths, including the affinity of 2 high-affinity binders, which I think I included here. Great, and that kind of wraps up what we've done on EvoDiff so far. Next, I'd like to move on to Dayhoff, which is our atlas of data and models for protein generation. Now, the creation and public availability of all these protein databases have directly enabled a new understanding of proteins. And this has been catalyzed by this parallel revolution in the open availability of data sets in this field. So if we think back to 1965, when we only knew the sequences of 65 proteins, today we have massive databases. The PDB contains hundreds of thousands of structures. UniProt contains millions of high-quality sequences. And now with Magnify and things like the Logan SRA, we have access to billions of metagenomic sequences. Now, this is the ability to understand and design proteins. This is the basis of many a modern PLM. And so to this end, our atlas includes a resource of both protein sequence data and these models as a centralized resource. Again, want to take another moment here to highlight some of these people involved in this project. Too many to name, and some of which I couldn't find photos. So includes people not voted here. This is multiple intern projects from the last year or so, as well as multiple of the technical staff contributions at MSR. Now, first I'll focus on describing the data, and then I will take the second part to talk about our models. Now, with our first data set, the goal is to expand the of natural protein sequences for protein language model training. This was mainly led by our awesome undergraduate, Kaylee, who did the majority of this work in 12 weeks, which was incredible. So these types of data, metagenomics, are obtained from extracting genetic material directly from environmental samples. So this includes things in extreme environments, mainly poop, but also things like volcanic regions or like really Arctic places. These metagenomic sequences are derived from samples, whereas a protein from UNIRAF is typically coming from an isolated, well-characterized organism. It's a part of a genome. Yes. Yeah, so people extract these genetic materials, they sequence them, and then they piece them together. So they infer like what the genome is by piecing them together. So to this point, it also results in like a lot of lower quality samples that might not be as reliable than a genomic example like from UniProt.
Mixture of things, and you take a sample of like a lake or in the natural environment, you have a mixture of different species, and you just do sequencing of this, and then you try to figure out what kind of sequences are inside.
Yes.
Yeah.
Yeah. Okay. Okay. So this results in data that is much more diverse and not structured, but these sequences can be much lower in quality. So we can create this combining, deduplicating, and reclustering sequences from 9 different source databases. This includes UniRef, but also databases like Magnify, Topaz, Merck, SRC, SMAG, and all these other names. These cover various organisms, so like microbiome, oceanic, soil, clinical. In total, this is about 5.7 billion sequences. There's duplicates and a lot of noisy data. And also because the total number of these sequences across these data sets exceeds the allowable limit of like modern clustering algorithms that are used. Famously, MMSeq is used broadly here. We have to perform this like multi-step clustering method. Kaylee actually happened upon this computational limit while trying to do this project. So first we cluster them at 90% sequence identity, followed by clustering the remaining representatives at 50% identity to produce this data set. This final data set contains 3.38 billion sequences total, 7 billion clusters. Now, 1.4 billion of these clusters or sequences are singletons, meaning that cluster is only formed by one sequence. We don't use these for training because generally you want a duplicate of something to enable some confidence, but we do release this data set. The remaining 243 million clusters we can use for training, and that will be the focus on the models that I present. Now, compared to UniRef, I want to point out that this is about a 16x increase in protein sequences, but also a 3 to 4x increase in the number of clusters. Now, to understand how these new sequences were impacting protein clusters, we took a look at the distribution of cluster sizes in the gigaref clusters versus the number of metagenomic sequences in each cluster. And the majority of them don't contain any UniRef sequences. That is, they're entirely metagenomic. Now, 13 million of these clusters have a mix of both, and none of them consisted entirely of UniRef sequences. This gives us evidence that metagenomic sequences are both augmenting UniRef by forming new clusters or augmenting existing clusters. Okay, and so now we can move on to the backbone ref dataset. This work was mainly led by our intern, Alex Lee, who's currently wrapping up his final year at USCF. UCSF, said that wrong twice. Now, beyond expanding the scope of the number of natural protein sequences, we came up with this idea of utilizing the success of structure-based models for the design of new functional proteins. As I've been mentioning, this structure-based approach remains state-of-the-art for functional protein design. And based on this, we hypothesized that we could distill some information about these successful structures directly into protein sequence information. Okay, so to do this, we develop a new pipeline to generate structure-based synthetic protein sequence data. We start by generating a large number of backbones. This is 240,000 backbones and took a few weeks of compute on V100s. We then operate a fixed backbone sequence design using a method called protein MPNN and inverse design sequences at multiple temperatures until we get about 2.4 million sequences. We then use a structure prediction model, OmegaFold, and look at the self-consistency between the RMSDs produced from the model and regenerated with this model. And then we select and analyze based on the lowest self-consistency RMSD, ultimately 240,000 sequence structure pairs. Now we can characterize the novelty of these generated structures by comparing them against the PDB database. So here we're looking at how unique these clusters are in terms of the number of backbone ref members that are contained in there. And of the 240,000 synthetic backbones, we get 83,000 clusters that don't have any natural members, meaning those structures are distinct from naturally occurring proteins. We can also kind of compare the novelty of these backbones by looking at their closest match to something in AlphaFold database and comparing this metric with a TM score. Here, a lower TM score is going to indicate higher similarity to a known protein. And most of our structures had high TM scores, indicating that they have significant structural novelty. Although you can see that this number quickly drops off as the proteins get larger. In all, we now have some confidence that backbone ref is giving us diverse and novel structures.
You say diverse, is it diverse sequences, but for the same structure?
Not diverse sequences, diverse structures. So with in mind that there are multiple of new clusters being formed, and the cluster would contain similar structured proteins. Yeah.
One structure for each sequence or multiple structures?
There are multiple sequences that we generated for each structure. Is that your question?
Yes. There is multiple combinations to each sequence.
Yes.
Yeah. So
Oh, I see what you're saying. So here because we're using these structure prediction models, we're typically capturing like a relationship of one sequence to one structure, or like many sequences to one structure, because we're not getting diverse structures in like a conformationally diverse way most of the time. And that's what I would expect out of a model like this, if that is more what you're asking. Okay. So with this in mind, that we have this diverse set of sequences, we now create 3 unique data sets by kind of downsampling data sets that contain 10 million sequences each. And we filter these by quality and novelty. So first, we just select sequences that we use an inverse design temperature of 0.2, so really low temperature designs. And then we filter them for self-consistency in the backbone and large TM scores, and we call these our unique, our quality, and our novelty backbone data ref sets. And we use these for various downstream ablations. Lastly, I'll also point out that we include data from Open Protein Set. This is a data set released by OpenFold, which now contains 16 million MSAs, which the prior release had contained 100,000 if you were listening earlier on this part of EvoDiff. So this is an order of magnitude increase since their previous release. Now, switching over to the modeling side, we release a set of 6 models that are trained at 2 different sizes, a small and a large, on these varying data sets that I just discussed. This includes a baseline model trained on UniRef 50 and 90, one trained solely on GigaRef, one on UniRef augmented by backbone ref. Keep in mind GigaRef includes UniRef sequences, and then bigger iterations of these models. And then this is a model that we cooled using some engineering tricks. Now, as you may recall earlier in this talk, I pointed out that autoregressive models outperformed our EvoDiff diffusion models on benchmarks that didn't require conditional generation, such as this FPD metric. Now, since the release of our paper, there were multiple advances in autoregressive models that made them attractive candidates for training our set of Dayhoff models, namely the rise of these hybrid state space transformers, which enabled long context lengths for multimodal inputs. And what this really did is unlock our ability to use a single model to handle both a single sequence input and an unrolled multiple sequence alignment. So now we could treat an MSA as an autoregressive task by unrolling it. This requires a 32,000 context length, so it's requiring a lot more power from the model. However, we still wanted that desirable capability of being able to condition with multiple directional contexts. And so as a clever way to incorporate this, with someone I was talking to earlier caught on to this, is we just train our model using two decoding orders. So the model sees sequences formatted from the N to C and C to N directions. Now, I don't have time to decompose all the results from these models, but at a high level, we can measure model performance over a few different metrics. One is model perplexities being evaluated over a held out cluster representative from UNIREF 50 and GigaRef, and the Fauché protein distance, which I introduced earlier in EvoDiff. And for both of these, a lower value is better. Now, as we expected, models trained on GigaRef or UNIREF will shift metrics toward those distributions, and synthetic data shifts us away from those distributions. And similarly, overall, we see that larger models are performing better than their smaller counterparts. Now we try to have some fun with the model and run a few ablations to see what we could do. This first experiment was using evolutionary conditioning to shorten proteins in silico. So we're not able to do any wet lab here yet. But we take unaligned Cas9 sequences, we format them in a unrolled fashion, and then we task our model with predicting the next sequence. And we do this with the goal of getting a shorter Cas9 that still contains interesting domains from other Cas9 proteins. Here's a few visual examples of proteins that come out that have high scores. And if we look at the domains that can be annotated for a natural protein versus a designed one, we see that we can retain some of these important domains involved in activity, but then also incorporate other notable domains that you might want to have in Cas9. And for this last in silico metric, we're looking at that scaffolding experiment where we want to be able to scaffold motifs in silico. Now, to do this with the Dayhoff model, we generate in one direction first. We go from N to C and fill in all the residues that the model wants to. We then go backwards and generate everything it wants in the opposite direction. There's an extra step in there. We generate everything N to C. I don't know why that's in there twice. And then we generate everything in the opposite direction. And what this does is allow it to infill or repaint in some of those generated residues or residues at the beginning that require that context of the later amino acids. Now, this plot is just looking at the score of each model on the scaffolding motif benchmark. It's taking the average number of successes for each problem and then averaging that out by problem number, where you want a higher score as indicating you do better on these examples. Overall, our models perform similar to how they do trend-wise on other experiments, and most of them. Here's just an example of two interesting protein regimes that we're able to generate for. One is this reductase active site, and the other one is a lactate dehydrogenase active site. Okay, and now we get to the final part, which was kind of the motivation for this in the beginning, is unbiased head-to-head comparison to examine the effects of model scale and training composition on the expression of different sequences. So we generate proteins from all of these models, and then we express each of them in two different E. coli strains, and a protein that expresses in either one or both of them is considered a successful expression. Now, looking at our small UNIREF 90 model, 8 of those 29 totally unfiltered designs that we send to the lab express. The addition of GigaRef results in 2 additional expressors. Our large model, our first large model that we test, has the same number of expressors, but we had less samples to work with here. This model, which includes MSA information, also augments. We get 12 out of our 29 expressors. And then interestingly, our small model, trained both on UNIREF and this synthetic augmented data set, gets the most expressors out of all of our small and large models, with 15 out of the 29 expressing. And this is cool because it was a totally unbiased experiment to just see what was going to express here. Now, finally, probably one of my favorite slides right now is we look at common in silico predictors that are commonly used to filter for downstream tasks. So again, we don't implement these for ours, but we look at things like PLDDT, the folding score, self-consistent perplexity, things we'd been using previously, homology, which many a reviewer has asked for before. And over multiple of these metrics, including a number I don't include on this slide, we see no correlation with these metrics and expressed versus not expressed proteins, which is quite interesting. And with that is the two projects we've been working on lately, EvoDiff and Dayhoff. We've summarized kind of our approach for unconditional, conditional generation, and we've done some interesting experiments on how data and scale improves expression. I'll also take this moment to highlight all of the interns' work and just like punch out that we're always taking interns every year. Applications are normally released between like September and December for the summer. They're 12-week programs. We love people that like come in with cool ideas or just like want to learn about protein language models or anything protein related. So please reach out if you're interested. And I'll take questions.